What's the point of such a complicated system? All right, you see that? Well, before we get to the point, let's look at how the molecule is held together. Non-covalent interactions, right? As we've discussed many times before. What I'm trying to show you here is that most of those non-covalent interactions, or at least some key ones, are salt bridges. Remember, positively charged side chains interacting with negatively charged side chains. So ionic interactions, charge-charge interactions, salt bridges. We're zooming in on a couple of those, and I'm just highlighting here in a few different ways how you might see where those salt bridges serve to tie together. In this case, the dimer, the individual dimers, right? The individual dimers, and it's the relative positioning of those individual dimers that changes as hemoglobin goes through its interesting gymnastics. So the tetramer is held together by salt bridges and other interactions as well. The great thing about hemoglobin, though, is that it is an allosteric molecule. It undergoes a change in the affinity of oxygen at different substrate concentrations. So if we haven't defined this term before, right, there's the protein and there's the ligand. Sometimes I'll say ligand, sometimes I'll say substrate. Ligand, a more generic term for anything that's being bound. Right, so the protein provides a binding site for a ligand that interacts with that binding site. Generally speaking, we'll say modification of enzyme activity through ligand-induced conformational change is our definition of allostery. Hemoglobin is not an enzyme. It does not change the structure of oxygen, at least not for any appreciable length of time. It just binds oxygen in one place and releases it somewhere else. So it's not really an enzyme. So modification of protein function then through ligand-induced conformational change. So there's that again, that idea of induced fit cropping its head again. Here's our definition: allos equals other, stereos structure. So we change our structure. Now there's a couple ways to think about this. Let's keep it simple. Again, let me restate hemoglobin's job. Hemoglobin is much more complicated than myoglobin because it has to pick up oxygen in the lungs, where oxygen is at high concentration, and it has to release it in the tissues, where oxygen is at relatively low concentration. That suggests that hemoglobin's affinity for oxygen is sensitive to the concentration of the ligand. Well, how does that work? So one way to conceptualize that is to think of hemoglobin as existing in two states. The tense state, drawn as a square here, the relaxed state, drawn as as a circle here, T and R. Tense being unable to bind oxygen, or less having a lower affinity for oxygen, put it that way, relaxed having a higher affinity for oxygen. One way to think about this, and this is the concerted model, is that the presence of ligand shifts us from the T to the R state. So if this is an equilibrium process, both of these might be considered to be existing in solution, not necessarily equally in solution, but the ligand pushes us towards another state. So high concentration of ligand pushes us to a relaxed, that is oxygen binding competent state. Conceptually, what that means is that high concentrations of ligand means that hemoglobin has a high affinity for oxygen. So in the lungs, its affinity is high. As the ligand concentration decreases in the environment, it switches to a T, which means that it pops off all of its oxygens. So this is a great method for regulating protein function. You can easily inactivate or activate a molecule just by the presence of some ligand. So if you want to alter your enzyme's activity depending on conditions, then you might choose some ligand that induces a conformational change that drives you towards one state or the other state. Again, depending on the particular requirements of a cell or other microenvironment in the organism. How does hemoglobin work again? So in this case, we're looking at again, relative saturation. Now we're representing it as Y. This is an oxygen binding curve. So in this case, we're representing oxygen concentration as the partial pressure of oxygen. and you could just as easily put theta and concentration of ligand here, right? In molar units, it doesn't really matter. What we see here for ligand is that we got a curve for myoglobin, right? Nothing interesting there, just a standard curve. We could fit that to our simple model and get a KD, right? Or in this case, you'd say a P50, 50% saturation. For myoglobin, that's PO2 of 2.8. This is conveniently also pointing out the relative concentration of oxygen in the tissues versus the lungs, and you see that it's 3 or 4-fold difference in the oxygen concentration, lower in the tissues, higher in the lungs. But what we'd also look at here is to say that myoglobin is totally saturated regardless of whether we're in the tissue or the lungs. Now we're not going to find myoglobin in any high concentration in our lungs, except in terms of the muscles that are used to contract and expand the lungs. But the bottom line is that the KD of myoglobin is way below the tissues or the lungs. So it's always going to be saturated at all oxygen concentrations. Not good if you want to transport and deliver the package. So let's look at hemoglobin here. We've got this sigmoidal curve. This is probably a little bit exaggerated so that you can clearly see. But what we see for hemoglobin is that, right, I think I have a little animation, so let's just do that. So myoglobin binds tightly but doesn't release easily. Good, that's what it needs to do. Hemoglobin has a low affinity at low oxygen concentrations. So we could imagine a case where what we really see here is a low affinity curve, and that's shown on the left here. If hemoglobin actually had two, you could plot the affinity or the binding for the two different R and T states. We'd see one state increasing very slowly. But then as oxygen concentration increases, we switch to the relaxed state, and then the binding curve of the purely relaxed state looks a lot more like myoglobin. But for the hemoglobin itself, we see this sort of S-shaped curve as we shift to higher concentrations. So hemoglobin has a high affinity at high O2, low affinity at low O2. Again, we can pick up, because we have high affinity, we can pick up 4 oxygen molecules in the lungs as we move to the tissues, the extremities, wherever. We see that we have a much lower concentration, and indeed we're below, or we maybe somewhere near the P50 for that T state. So we can drop our oxygen in the tissues. We'll refer to this as cooperative oxygen binding. So there's a cooperative change in the behavior from T to R, and that cooperativity gives rise to this S-shaped curve. We can quantify this as well, and now we introduce a new model that has this guy here, the n. And when we write an equilibrium constant, then the n becomes an exponent, right? So here's an association or dissociation. Let's stick with dissociation for our purposes. The dissociation constant now has this exponential feature that, again, that allows us to quantify the cooperativity, right? The interaction between those sites. That's important. So we can use that n as a metric of just how cooperative the hemoglobin monomers are with each other. How does the binding of oxygen at one site affect the structure of the other sites? Well, we can do something, and we'll play with this in class a little bit. We can create something like a hill plot, and this is just a reorganization of that. Remember what I showed you before was the fraction bound theta is just a combination of the ligand concentration and the KD. So we'll work on how to derive this equation ourselves in class, but it's a very straightforward little bit of algebra, and we can express the log of the fraction bound over 1 minus the fraction bound in terms of the ligand concentration and the KD and this hill coefficient that gives us a sense of the relative cooperativity. Now, this is really something that one might do for the easy analysis of n. So if you look at this, this looks like more or less a linear function now. Even though there's all those logs in there, this is more or less a linear function. And what we got here, if we looked at log of the theta over 1 minus theta versus the log of the ligand concentration, we have an upper bound that might be for the R state, right? The affinity for the R state, that's the T state. But because R and T actually coexist and are in equilibrium with one another, N will define how we shift from one to the other and where. So as we go from low concentration to high concentration of ligand, we switch states. And really what we would see if we were to look at this kind of data, we'd see a line at low affinity. We reach our trigger concentration and we switch to the high affinity state. That switch defines N. So here we have for the R state we have an N of 1, for the T state we have an N of 1, but as we move from T to R, our N increases. That is a reflection of how cooperative the change is between T and R. Okay, this is a figure from one of the articles that I gave you. Let me just state that Doug Barrick is now a full professor at Johns Hopkins. He was a postdoc in the lab in which I did my graduate work, and he helped train me in terms of how to make mutants and how to purify protein, how to be a scientist. So I really am pleased to show some relatively old work of his. But those of you that are interested in studying biophysics, apply to Johns Hopkins. He's a great guy to work for. In any case, what we're looking at in this case is, all I want to point out to you is the Hill coefficient of hemoglobin and myoglobin. So this is that N that you would get for if you were to fit binding data, oxygen saturation data to the Hill equation. And for hemoglobin, wild type hemoglobin, it's obviously greater than 1. That tells us that hemoglobin is pretty cooperative. That is to say, all of those active sites seem somehow to switch from a low affinity to high affinity state at some fairly low oxygen concentration. Myoglobin, of course, has a Hill coefficient of 1 because it's just a straight line when you plot the data as log, and there's obviously no conformational change and no switch in affinities. We'll talk about these mutants of hemoglobin in class. I really just wanted to point out to you those Hill coefficients. As well, I wanted to show you some actual data. This is the original data from the original Hill paper back in 1910. Now it's actually, if you look closely, he's taking somebody else's data, Barcroft and Camus. I didn't go back to their paper, but what he's done is fit this is hemoglobin binding data, fit this data to his own model, his own Hill equation, to look at the relative cooperativity of hemoglobin at different ligand concentrations here. So he's done a big faux pas in my mind. He hasn't labeled his axes. So, but then again, that's hemoglobin saturation and oxygen concentration. You can see that, you know, his binding curve doesn't really capture what we would say is cooperative. In fact, if I looked at this, I'd say there's no particular reason, just gazing at the data, to decide that this is a cooperative system. There's no sigmoidality to the curve. It looks like a straight up binding curve, and I think you could probably fit it easily either way. But in any case, this is the data. These are the data that he started basing his models for cooperativity upon. Let me say something else too, though, that when you've linearized your data like this, you can fit it easily to this version of the Hill equation. But you can write the Hill equation different ways that are actually. Let me zip all the way back to here. Blah blah blah. Let's just Oh, sorry, we're gonna go way back. Right, you can basically fit your binding data to this kind of expression if your data takes the form of one of these rectangular hyperbola in this case. Or as you can see in Barrick's data over here, a clearly cooperative change going on, an allosteric change in the affinity of hemoglobin happening there as we increase oxygen concentrations. Part of this is because it's really hard to maintain oxygen concentrations at low levels. So you can see that these guys have nothing below 10%. At least Barrick was able to get a few points below 10% oxygen concentration. We won't talk about this now, but he's also in much nicer data, allowing you to go to much lower dilutions in terms of substrate concentration is this guy who the abbreviation for which some kind of cyanide ion, I can't remember what. We'll talk N-butyl cyanide, I guess. Uh, he's got other saturation data here for his constructs in the presence of this oxygen alternative, if you will, and you can still see the cooperativity quite nicely, and you can fit that to the Hill equation as well. And it's these kind of data, right, nonlinear data, that we're getting these Hill coefficients from. In any case, there's a variety of ways to fit your data. You can fit it using using linearized data. You can use it. You can fit it using sort of much more complicated nonlinear regression type analyses. But you get the sense now of relative cooperativities. I think the triangles here are myoglobin binding data and some of his mutants versus wild type hemoglobin. Very clear in this actual experimental data that there's a low affinity state and a high affinity state, and we go through the switch somewhere around partial pressures of oxygen on the order of 20 millimeters of mercury. How does it all happen? Iron coordination by the heme is the key. So again, we have this ferrous iron in the middle of the heme. That's a hexacoordinate iron, so it needs 6 coordination elements in order to be fully bound. And heme provides a 4, right, through those paroles. Proximal histidine provides another. So there's the proximal histidine, histidine 93, right? And then oxygen can provide a final ligand. Well, you might have noticed in the last slide, in that first oxygen or myoglobin slide I showed you, that the binding to that proximal histidine actually distorts the heme relatively significantly, pulling the iron atom towards F8, right? Helix F in that original globin helix naming system. So that's the whole structural story of how hemoglobin can transmit a conformational change after ligand binding to its partners. Deoxyheme allows the iron to pucker, as I've said. That you can see the distortion quite clearly here in the T state, right, interacting strongly with that histidine. But when we bind oxygen, that 6th coordination site draws the iron back into planarity with the heme. So that heme is covalently linked to the histidine. That means that the F helix moves along as well. So we have a conformational change that occurs as we move from the T to the R state. Oxygen binds, pulls us in, and that you could imagine then can be transmitted to the rest of the molecule. Remember that this helix F doesn't exist in isolation. It's interacting with other helices in hemoglobin, and so that simple change is transmitted to the other can be transmitted to the other molecules in the tetramer, the other monomers in the tetramer, to induce a change in the affinity of the other sites. Motion of the F helix repositions alpha 1 beta 1, alpha 2 beta 2 interface, resulting in increased oxygen affinity at the remaining 3 binding sites. So this oxygen, let me say first of all that oxygen is an example of a homotropic allosteric effector. So there's a little bit of allostery terminology. It's an allosteric effector because it changes the relative affinity or changes the function of the molecule, hemoglobin. We call it homotropic because it's the actual ligand molecule, the functional molecule that hemoglobin is designed to interact with, if you will. But this term becomes a little bit slippery. But let's think of it as binding at hemoglobin's actual active site, homotropic allosteric effector. As opposed to heterotropic effectors that we'll encounter in just a few minutes. But I think when I say this homotropic versus heterotropic effectors, when I say that's a little bit slippery of a concept, well, what's the right binding site, right? There might be other binding sites. Hemoglobin acts on other molecules in ways that are maybe not equally important to oxygen binding, but are also very important to biology. So which one do we define as our actual real binding site. And it's fair to say that oxygen binding and release is the job of hemoglobin. So since oxygen binds, since oxygen is the ligand, it's a homotropic effector, and other ligands alter the behavior of hemoglobin with respect to oxygen affinity. So those are heterotropic effectors. Back to structural basis of hemoglobin cooperativity. So I mentioned a model at the beginning of this discussion, which was a concerted model. That is to say, the hemoglobin exists in the tense state or the relaxed state, and those two states are in equilibrium, and the ligand shifts us to one end of that equilibrium or the other. There's also a sequential model of allosteric interactions or cooperativity that is more of an induced fit kind of model, and that would be each as an oxygen binds at one site, structural changes are transmitted via induced fit model or an induced fit mechanism to neighboring sites, and as more oxygen that changes the affinity of the unbound site, but as oxygen binds at the new site, that again transmits structural change to the other free sites. So there's a sequential change in the structure that takes place as the ligand binds. Formally, the concerted and the sequential models are different, and they seem to have different implications. In practice, it's very difficult to distinguish those, and well beyond the scope of this class to try to think about how we might distinguish those experimentally. They both have different attractive features. On the one hand, the concerted model allows us to think about the R to T transition as a chemical equilibrium that's shifted by the presence or absence of ligand. On the other hand, the sequential model allows us to think about ligand binding inducing changes in the structure and transmitting those changes to other sites. So they both are attractive. Let's be able to formally think about the two as distinct, but the distinction is a little bit academic since we don't have good ways of distinguishing between them. See animation. So I have an animation of this change for you. So let's switch to, yes, here we go. So deoxy, oxy. Watch that blue oxygen molecule, right? And what it does to the relative position of the proximal histidine and to the rest of this protein as well. So we've got a tetramer here. The one that we're focusing on is the pink one, but you can see that the whole molecule is changing as well. And I'll draw your attention, and I'm not sure that I'm drawing your attention to exactly the right. There's a lysine, clearly a lysine. Big change, oxy, deoxy, right? Salt bridge, no salt bridge, salt bridge, no salt bridge. Here again, right? There's a glutamic acid, right? Shifting alliances as we go from the oxy to the deoxy state. Here, note the hue. So overall, you might think, well, the structure globally doesn't look to be changing that much, but look at the size of this, the motion here, right? Maybe 6 or 8 angstrom change in the relative position of that lysine. So big changes, and you could easily imagine that there are oxygen affinity changes that happen. So let's think about one oxygen binding at one oxygen binding site, inducing a conformational change in the whole molecule. So suddenly you've gone from 4 sites empty and low oxygen affinity to one site being filled, but all the others being switched to high affinity. What are they going to do now? Bind oxygen. Okay, back to our story. Hemoglobin undergoes a conformational change, changes its affinity for oxygen. Other ligands attenuate activity. So 2,3-bisphosphoglycerate is a heterotropic allosteric effector, doesn't bind at the heme, doesn't interfere physically with oxygen binding in the sense that it occludes the heme or something. Instead, it binds at the interface of the subunits. And furthermore, binds selectively to the T conformation. So reduces the relative affinity for oxygen and and keeps us in the low affinity state. Here you can see close up of bisphosphoglycerate and the salt bridge interactions that it's forming with various side chains. So very complex, extensive electrostatic interactions holding bisphosphoglycerate in place. And I should say that most cells, adult erythrocytes have about 200 millimolar bisphosphoglycerate. I want you to think about the effects of changing the bisphosphoglycerate concentration, and we're going to work on problems related to that in class. So the negative heterotropic allosteric effector being an effector that attenuates or decreases the activity of the protein or the function of the protein. There are plenty of other heterotropic effectors. Let's think about the Bohr effect, which was named for the father of the famous physicist Niels Bohr. I don't know what his first name is. 0 extra credit points to the person that can come in with Niels Bohr's father's name. Anyway, noticing the effect of pH on oxygen affinity of hemoglobin. These heterotropic effectors, they affect the binding, also attenuate the oxygen affinity of the molecule, and they also serve to remove some poisons. So changing the pH, for example, and removing the respiratory byproduct carbon dioxide from the system. So first of all, as carbon dioxide is released, for example, from the Krebs cycle, the tricarboxylic acid cycle, stay tuned for your metabolism if that doesn't mean anything to you. The hydration of that, right, to carbonic acid results in a change in the pH, a lowering of the pH. The lowering of the pH favors T-state salt bridges. I want you to think about why a lower pH might stabilize the salt bridges that lock us into the low affinity state. Furthermore, binding of carbon dioxide to hemoglobin's termini, so the piggyback ride for carbon dioxide to leave the tissues on hemoglobin for exchange in the lungs, is at the terminal alpha amino group and of course hemoglobin has 4 of those, so we can carry out 4 carbon dioxides for each hemoglobin molecule. But as we bind that CO2, we release still more protons, and that results in the further tightening, right, or further favoring of the T state. So we have a whole complicated system for lowering the affinity of oxygen in the tissues, right? All of these effects are gonna be at their highest in the extremities, where hemoglobin needs to have a low affinity. So we switch to the T state again in the tissues, and then as we exchange away, right, our CO2 in the lungs, and as the oxygen concentration is increased in the lungs, we switch back to the R state to pick up our hemoglobin again.